Yeah, so Casper just told you basically how the procedure works and how you select these ROIs and shine UV lights on the ROIs, which leads to the segregation of these probes that were once attached to the RNA that you're measuring, and now ends up into these wells, which then goes onto the sequencing machine. And what happens after that is that when the sequencing machine finishes up its sequencing process, it makes a number of actually BCL files, which are then transformed into FASTQ files. And you probably, most of you know how these FASTQ files look like. They are just basically plain segments of sequences that comes out of the sequencing machine. And then the trick is to get these sequences into counts of gene expression. And once you have a gene expression count, this is produced in files called DCC files for the GeoMx platform. Well, then you have an option of putting these count files into the GeoMx machine or computer attached to the machine again, and then you have like a clickable environment where you are able to look at your data and do stuff with it, do differential expression, etc. But in our approach here at MoMA, we are very much into reproducible research. So we like instead to take out these counts and work with them in a compute environment such as the R language. For doing this, we are working with a workflow that is depicted down here, where we do all the things that we basically need to do in order to analyze the data. And that includes some quality control things and filtering and normalization, and finally an analysis of the data. And this is something that we do in a close collaboration with the investigators of the experiments. So I'll talk a little bit about the process of going from the sequence files to the count files, and then I'll go through what is the content of this analysis procedure. So what we do at MoMA is that we make an analysis report in the HTML format, so you can look at it in a browser, and I'll go through what is the content of such a report. So first, let's see what happens when we go from sequences to gene counts. So we have our regions of interest, or our segments here, and what happens is that the complementary DNA sequence is targeted to the RNA, and then we have this photocleavable linker, which is attached to a DNA oligotag. And the UV liberates this DNA oligotag, and it goes to the sequencing machine. This DNA oligotag, or actually the whole sequence here, consists of first an antisense to the target, and then we have the photocleavable linker here, and then you have a reverse PCR primer binding site, and then you have, and this is actually what comes out of the sequencing machine, a 40 nucleotide unique molecular index, and then another index beside it. And this index, called the RTS ID, is a target specific index that kind of points back to what was it that the probe actually annealed to. So this is actually the information you need to sequence in order to know that this probe attached to an RNA molecule in your segment. So once you have the output of your sequencing machine, it looks like this, and you have your sequence here. You have the UMI in blue and the RTS ID in green here. So the next thing that happens is that a software called GeoMx NGS pipeline takes the FASTQ file that we have here and an ini file that is produced by the instrument. So this ini file has a translation from the RTS ID that was reflecting the probe that bound the target, and then an ID that tells us which probe is it and what gene is that. And then the software counts up how many times it finds this RTS ID and with unique molecular identifiers, and then it counts up how many there are of these. And for instance, for this particular one we have right here, RTS number 33,912, it counts up that it saw this 16 times in this particular segment. Then for each segment we get one DCC file that counts up all of the probes corresponding to all of the genes that are on the set that we are using in the experiment. Okay, so that was how the count matrix or the count files are made. Now I want to talk a little bit about how or what we do when we have the count files and start doing further analysis of the counts. So at the moment, what we do is we produce a project analysis HTML report, and this is a report that was the content of which was originally developed by NanoString. So here at the left of the screen is the original report that is based on a kidney data set that is kind of a test data set that you can download from NanoString's website. So we are using basically the code from this report and modify it along the way and try to generalize it and adapt it to the experiment that we are currently working with. So our emphasis is to kind of reuse as much code as we can, and as I said, try to make it work in a generalized manner for all the projects that we run through our pipeline. One thing that we like about this report is that all the plots and all the analysis that is going through, we kind of have the code included in the report, so the receiver of the report can see exactly what has been going on, and they can also, in fact, reproduce the whole report by themselves if they have the data. And in this way, they can also easily, if they are a little computer savvy, change the code, change, for instance, titles of plots, etc. And in this way, hopefully, the report will be useful for the end user. So basically, the different parts of the report are some QC and some filtering and normalization, and finally a differential expression analysis. So I'll just go briefly through some of these elements. So for instance, one of the first things that we see in such a report is a project overview in the form of a Sankey diagram. So this is where we can kind of have an overview of the project here. It is exemplified by a mycosis project, and here the main thing about the segments were whether they were whether the segments were procreating or not, and we can see how the segments are divided into P and N here, but they are also divided into the different slides and the different cell types and the different patients. So this is a nice way of getting an overview of the project. Another thing we look at are some quality control measures. So we look at whether the reads in each segment are trimmed and stitched and aligned appropriately, and we look at the negative probes, whether they look as they should, and we look at things like sequencing saturation. We plot things like how many genes that are detected in what number of segments. Plots that looks like this. Here it's for our bladder TMA project that Kasper talked briefly about. We look at the raw counts of the segments, and we make a normalization, usually a third quartile normalization, and look at what the counts look like after this normalization. And then we do some unsupervised analysis where we can see if the gene expression data segregates the variables that are of interest in the project. We also make some hierarchical clustering of some of the most variable genes, and finally, and perhaps the most specific thing for each project is that we do a differential expression analysis. And here there is, of course, some work between the researchers and the bioinformatic part in finding out what types of contrasts or model we want to be looking at. So here on top we see volcano plots, plots of gene expression fold change, and the way you should kind of read these things is that you have, for instance, at the bottom here, you have basal layer cells versus CD4 type of cells. So it basically means that the segments containing basal layer cells are contrasted against the ones with CD4 cells. You can see here, for instance, we have a gene here, LTB4R, which is increased in the basal layer. And when we then plot the expression of these genes for all the segments in the experiment, we can see how the basal layer has a higher level, regardless of whether there was progression or not, than these CD4 MA cells that have a lower expression here. So these are the types of things that we include in our report. And in conclusion, so for the GeoMx projects that we run at MoMA, well, we use a stable universal pipeline for generating our counts. We like to run these reproducible analysis workflows, and we obviously collaborate with researchers on how we design the models for expression, and then we make these customized reports that we hope are usable and at least have reusable code so that you can reproduce what was done.